Okay, so let's get started. Thanks everybody for joining. It's really wonderful to have you here. And thanks also Chris and Maris, I hope I'm saying your name right, for joining. Hope you're all staying warm on this snowy day in Calgary. And if you're joining from elsewhere, it's really great to have you here. Thank you so much for kicking off Precision Health Week. We do have a few other events happening this week as well. So if you'd like to join on Tuesday, we have a session being delivered by 4 of our highly skilled recent graduates, precision health projects in different healthcare organizations. These are different types of projects, quality and safety project, an equity project, a curriculum project, and a pharmacogenomics project. So please feel free to check that out. On Wednesday, we're going to have the director of the Precision Health Center in the Maritimes, brand new center, who's going to be joining us along with Lena Desai, and they will be talking about how to establish a precision health center. And on Friday, we're really pleased to be joined by Chad Saunders, Olex Oskiewski, and Jo Louise Huck, all doctors who will be talking about how to advance innovation and entrepreneurship in healthcare. Doctors meaning PhDs who are experts in innovating and entrepreneurship. So please feel free to join the URL on the screen there to get more information about how to register, or you can use the QR code. And thanks again for joining. Maya, I will pass the mic to you.
Excellent. Thank you, Ivy, for this introduction and for kicking this series off. So I would like to now welcome you here today for the 2 excellent talks on the changing landscape of precision oncology that will be presented by Drs. Erik Nohr and Pinaki Bose. So first, to introduce Erik, Dr. Nohr is a molecular pathologist with Alberta Precision Laboratories and an assistant professor at the University of Calgary. He is board certified in anatomic pathology and molecular genetic pathology, and he completed anatomic pathology residency in Calgary, molecular genetic pathology fellowship training at Stanford, and pediatric perinatal pathology fellowship training at Alberta Children's Hospital. He's a member of the Alberta Molecular Pathology Program, and he's a scientific lead in the South sector, where he oversees clinical molecular assay planning, development, validation, implementation, quality assurance, and quality improvement of neoplastic disease. His research focuses on inter-assay technical comparisons and novel assay designs to optimize molecular profiling of cancer patients. He's also the provincial educational lead for the Alberta Molecular Pathology Program and is actively involved and passionate about teaching. So without further ado, Erik, please go ahead and tell us more today.
Thank you for that introduction. Can you see my slides?
Yes, looks good.
Great, thank you. All right, so hello everyone, and nice to meet you. So for my part of the talk, I will talk about some more general principles in the field of precision oncology, and then afterwards, Dr. Bose will go into some more details about some specific hot topics and emerging technologies. So I like to start with these 2 images because I think they highlight popular perceptions of precision oncology, which may not be entirely accurate. On the image on the left, we see a patient reviewing the letter-by-letter sequence of presumably their genetic code with a physician, maybe their family doctor, maybe their oncologist or medical geneticist. But in reality, the information that patient-facing physicians, such as oncologists and geneticists and others, act upon has to be highly digested in the lab first. So when we do sequencing of, for example, a patient's cancer, there's a pipeline for analysis of that data, and there's key involvement of medical lab technologists and bioinformaticians and molecular professionals who parse that data and extract the clinically meaningful data and put it into some type of report. And then that's what is acted upon. So here I've put an example of an ALK fusion identified in a lung cancer, which is targetable. On the image on the right, we have a patient holding a pill with their face on it, with the implication being that that patient's treatment for their cancer is tailored specifically for that one patient. Now, it is true that in precision oncology, we test and develop an understanding of the molecular biology of a patient's cancer in order to more effectively direct their treatment, but it's not quite at the level of individual patients yet. Rather, it's more so that we're dividing patient cohorts into smaller and smaller subpopulations with specific genetic features so that we can use treatments that are more effective for that category of cancer patients. So taking a step back, how do we define these terms? Well, so the overarching umbrella is precision medicine, which is the direct application of big data, molecular biology, and computational tools in the clinical context. Precision oncology then is one part of precision medicine that is related to treating cancer. And this includes developing and implementing treatments that target the molecular characteristics of an individual's cancer. And as I mentioned in the last slide, this is a further substratification of cancer patient care in order to apply treatments that are more tailored to the molecular biology that go beyond surgery, chemotherapy, and radiation therapy that have been the traditional three arms of cancer therapy for many decades. And so the field of precision oncology is characterized by a rapidly increasing clinical need and increased tools for characterizing individual patients' cancers. On the left is a diagram of FDA approvals for targeted therapies that are based on genomic biomarkers over a couple of years span. And this is just meant to convey that there's a lot of them. And in the years since this diagram came out, there's even more. If anything, the pace is increasing. Meanwhile, though, what helps labs is that there are rapidly dropping sequencing costs. So this is the cost of sequencing compiled from the NIH, cost per human genome, with cost on the y-axis on a logarithmic scale and time on the x-axis in years. And so we have Moore's law here, and the drop in sequencing costs have actually outpaced Moore's law. You'll notice there's a particularly large drop around 2007, 2008, which is when next generation sequencing came out as a major technology. And in fact, over about 10 to 15 years, there's been a drop of about 100,000th of the cost for sequencing. So this is a big picture of who's involved in precision oncology. There are many stakeholders. Ultimately, everything is centered around the patient. And this work begins in the preclinical research community where new biomarkers are assessed and developed. And then Health Canada and Canada's Drug Agency play critical roles in evaluating the safety and efficacy of new drugs that come out. Pharmaceutical companies and molecular biology reagent vendors are very deeply invested and have many have a long reach and deep ties into all aspects of this development pipeline. And then ultimately it all comes back to patient care, which includes physicians, non-physicians, and critically the patient themselves. Focusing on the patient themselves and who's involved in that cancer patient's care, I've laid out here physicians and non-physicians that are involved. So physicians, I will start at the bottom and go clockwise. Pathologists play a critical role in diagnosing cancer. At the start of every cancer patient's journey is the diagnosis of what their cancer is. And pathologists examine cancer tissue under the microscope and perform various ancillary techniques to further characterize the biology of the tumor in order both to give it a name and to collect clinically relevant information about it. The radiologists play a critical role in imaging tumors, which is important both for diagnosis and for follow-up and for planning interventions such as surgery. Now, if you're a cancer patient, you hope that a surgeon is very involved in your care because the best kind of cancer to have is one that's small enough to cut out and cure. Surgeons do cure many more cancers than oncologists do. But oncologists become key whenever there's a need for medical treatment of the cancer, whether it's chemotherapy, radiation therapy, or targeted therapies based on the molecular biology of the tumor. Also, medical geneticists play an essential role for any patient who may have a hereditary cancer predisposition. For example, hereditary breast and ovarian cancer from BRCA mutations. And that has huge implications, not just for the patient, but also for their family members about monitoring for disease or prophylactic surgeries. Now, a number of non-physicians are also involved, including molecular geneticists who are PhDs that have clinical training to sign out molecular reports, medical lab technologists and lab scientists who are in there doing the pipetting and actually performing the testing, bioinformaticians to process that data, and then patient-facing non-physicians such as nurses, genetic counselors, and pharmacists. Just as there are many people involved, there's many types of biomarkers or substances that we look at in precision oncology. And so these include all the different types of omics. So there's genomics for DNA, there's epigenomics for methylation, transcriptomics for RNA, proteomics, metabolomics, et cetera. These are all just different ways to characterize a tumor. And why do we care about all these different omics and these different biomarkers? Well, it's because they provide information that can help to diagnose a patient's cancer or determine how bad it is, so that's prognosis, or predictive markers that predict responses to targeted therapies, or predisposing mutations, by which I mean mutations that may be present in the patient's germline or constitutional genetics that give them a cancer predisposition. Layering further complexity onto all these different utilities for biomarkers is that they're most commonly cancer specific. So this diagram is laying out some of the key genes, definitely not a comprehensive list, but some of the key genes across cancer types of different organ systems. There are rare exceptions where there's tumor agnostic drug approvals, meaning that it doesn't matter what cancer type you have. If you have this type of mutation, then there's a treatment for it. These include NTRK and RET fusions and immunotherapy. And we should perhaps not be surprised that different cancers have different mutations. Even though all cancers have certain core things that they need to accomplish in order to be a cancer, which we call cancer hallmarks. These are things like, for example, being able to invade and metastasize, being able to have replicative immortality, so divide essentially forever, and many other features. But each different cell lineage has different pathways and different genes it expresses, and so correspondingly, mutations in different genes are important in different cancer types. An example of a major success story in precision oncology is EGFR mutations in lung cancer. So EGFR is a receptor tyrosine kinase, in other words, an RTK. And so there's a variety of different RTKs. And so these are cell membrane proteins that sit on the surface and have an extracellular ligand binding domain. And when the ligand binds, that triggers a change within the intracellular tyrosine kinase domain to become phosphorylated and then signal downstream through multiple effectors to ultimately confer some of those hallmarks of cancer. And so EGFR is one of those RTKs, and activating mutations in EGFR occur in about 20% of lung adenocarcinomas, which is a particular type of lung cancer. And it turns out that these EGFR mutations are really key to the clinical management. And this is work that goes back multiple decades to develop tyrosine kinase inhibitors, TKIs, that specifically inhibit EGFR and prevent that downstream signaling and cancer hallmarks. And so comparing to standard chemotherapy from decades ago, looking at the overall survival in months, you see that there's a huge increase. Advanced stage lung cancer is usually a death sentence within like less than a year. Like outcome is measured in months. You can see here about like 6 months. Whereas if they have an EGFR mutant lung cancer, then you can treat with these targeted therapies and then they survive for years. So that's a huge difference. Now, it's all well and good to say that EGFR mutations are critically important in lung cancer, but which EGFR mutations? There are many different variants that can occur in EGFR or in any gene. And so when we do sequencing and we find those variants, the sequencing data itself doesn't tell us whether that's actually clinically meaningful. We have to look at a variety of different features to decide whether that will actually be clinically meaningful and should prompt a change in management. There are some variants that have a large genetic effect, like deletions, frame shifts, nonsense mutations. These ones we can essentially infer that they'll impact protein function just based on the type of mutation. Otherwise, Making this distinction comes down to a lot of clinical, pathologic, and molecular correlation for various features, including the frequency of that variant in the population, conservation at that locus, predicted and cell culture or animal model studies, and a variety of clinical information. And so this is the type of work that molecular professionals are doing routinely. When cancers are sequenced and we find things, we have to determine what they actually mean. And so there's a whole potpourri of different databases that continually evolve and that each cover different parts of the puzzle, different aspects of that variant annotation that we need to perform. And so we become very familiar with these databases and what they provide and how we use them to arrive at clinically meaningful information and process the sequencing findings into that type of high-level summary that I showed at the beginning in that report that the clinician can actually act on. Wrapped into all of this, there's a continual question of how much should we actually test? It's very easy to test more genes. Doing whole genome or whole transcriptome sequencing is actually the most technically simple kind of assay because you don't have to do any kind of selection for your genes of interest. But the challenge is that the genome and transcriptome is a big space, and sequencing samples cost money. And to sequence it well enough, even when you have, say, poor quality or samples that have a lot of background normal tissue, you need a lot of sequencing reads, and sequencing reads cost money. So I like to compare it to spreading butter across bread. If the bread is like the genes and the butter is like your cost per sample. This is why panels are so prevalent right now in clinical testing, is because if we do a narrower panel, we can contain the costs while still providing the all the relevant clinical information. However, that being said, there is this continual evolution in molecular test methodologies, where in the past, we didn't have these comprehensive panels, so we needed to do single biomarker analyses step by step. But that was creating challenges because every time you do a new test, you use some more of the tissue that, that was acquired from the patient. And so biomarkers or biosamples become very limiting for that. Current state is NGS panels are the most common method to provide clinical testing for cancer patients. And so there's a variety of different panels that are tailored to specific cancer types. So for example, you might have a lung panel or a colon panel, et cetera. However, those panels are increasingly being consolidated into larger panels, which may either be pan-cancer gene panels or can progress to whole genome sequencing or whole transcriptome sequencing to just cover everything all at once, get it done with. Single test on a single sample. So overall, over time, we have a decrease in methods per sample and an increase in data per sample. But as we scale up to higher and higher gene lists and panel content, we always have to maintain robust clinical testing. It does no good to test the whole genome if we miss that EGFR mutation and shave many years off a patient's life. So we need to make sure that the specimen is a good enough quality, that the testing is performed in a clinical lab that's accredited, so that there's regular checking on the quality of the lab, that it's meeting required standards. We need a diagnostic test that, is validated or verified, meaning that their performance metrics have been determined, so that we know what the accuracy, the precision, et cetera, is. We need a pathologist or a molecular geneticist who is certified and licensed and qualified to interpret this data and sign out reports. And once we have all those things in place, then we can have an accurate and reliable result, and we can avoid false positives and false negatives. Also tied into the field is ethical issues that arise as we test more and more things. There's always questions about how much information should the lab return? How much evidence does an oncologist need to actually act on a finding? We find variants all the time that there may only be 1 or 2 papers out there that did a little bit of cell culture work to determine that there may be a gain of function. So should the oncologist use that to treat the cancer? What should be done with potentially germline pathogenic variants that are identified on cancer testing? This happens all the time. It's probably around 20% of cancers we test that we find something that could be germline that we flag for potential medical genetics follow-up. And then that raises the question as well, should there be return of incidental findings and sequencing raw data to patients? And what are the privacy and communication risks, and how do we get patient consent to participate, for example, in clinical trials? So to summarize my part of the talk, precision oncology is a rapidly evolving multidisciplinary field. There's an ever-expanding list of diagnostic, prognostic, and predictive molecular biomarkers that is increasingly used to guide patient care across all types of cancer. And the identification and interpretation of those biomarkers depends on navigating numerous molecular databases that each give different parts of the puzzle that we use to interpret the data generated for any individual patient's cancer. There are many nuanced ethical issues in the field, and clinical molecular labs play a vital role in cancer testing through developing robust, reliable assays with documented, validated performance so that we can be assured that when we test for something, if it's present, that we will find it. So that's all for my part of the talk. So I believe we're taking questions at the end. So I'll pass it over to Dr. Bose.
Thank you so much, Erik.
Thank you.
This was really great. And I will now introduce Dr. Bose. He is an associate professor in the departments of oncology, biochemistry, and molecular biology, and pathology and lab medicine at the University of Calgary. He holds multiple leadership positions within the Cummings School of Medicine and the Arnie Charbonnet Cancer Institute, including scientific lead for the Precision Oncology and Experimental Therapeutics, or also known as POET program. Pinaki is Director of Tumor Biology and Translation Research for the Olsen Research Initiative and scientific lead for the Calgary Global Medical Education, or CGME program. Dr. Bose leads a pan-Canadian team of researchers investigating the genomics underlying head and neck cancers. And at the Bose Lab, located in Arthur Child Comprehensive Cancer Center, his team leverages advanced omics technologies, including spatial transcriptomics and functional assays, to uncover the drivers of cancer metastasis, immune evasion, and resistance to immunotherapy. Dr. Bose's research has been published in high-impact journals, contributing valuable insights into cancer biology. So without further ado, Dr. Bose, please go ahead and tell us more about changing landscape of precision oncology. Oh, Pinaki, if you can, I'm not sure if you can make the slides a little bit bigger, or I'm not sure if this is just the way I see them.
I'm doing like I'm screen share. I don't know. Is there any? The slides look small?
Yeah, it looks small for me. I'm not sure if Ivy's still here. Oh, now it's so if you can now do the presenter mode.
Yeah.
Oh, and then it shrinks again. I'm not sure why.
I think it's okay. I'm not sure if you move your mouse a little bit. I did see a percentage in the bottom right corner of your screen, but I don't see it now. So I think it's okay, Pinaki. We may not be able to see any fine text or diagrams. We can see it at.
I don't know why this is the case. Just let me just see.
Okay.
It doesn't give me anything that would change. I don't know if.
We can still see it. It's just a little bit smaller than the full screen. So feel free to proceed and
Screens. So now, is it good or?
It looks good. And if I try to put in presenter mode, yeah, it still goes back to small, but I think it should be okay.
Okay. I don't know, like I'm just doing presenter mode and it's big on my side. There's something, maybe some glitch somewhere. Anyways, I'll start, and if people can see, please tell us, and we will try to figure something out. But yeah, today, so you heard Dr. Nohr talking about basically precision oncology and why we are doing precision oncology. I wanted to get a little deeper into the practice of precision oncology. This was already discussed by Dr. Nohr, so why there has been a paradigm shift in how we treat patients. So oncologists or any clinician wouldn't want to treat patients non-precisely. Like precision medicine has always been the underlying concept when patients are being treated. We always want to treat patients precisely. But what has changed in the last 50 years or so, or maybe in the last 20 years more rapidly, is that a lot more data has become available, and all of the data taken together has led to more precision in treating the patients. So this is what we used to do, a one-size-fits-all medicine. Then we went into stratified medicine, where essentially patients are grouped based on disease types, essentially their demographic profiles and other risk factors, comorbidities, etc. And they are treated separately based on those. But then as we practice more and more precision medicine, we are stratifying patients based on their genomics and other omics profiles. We also integrate a lot of clinical data into it. We don't throw the baby out with the bathwater. We use all the clinical data because that is very important. Without the clinical data, the genomics doesn't make sense, or the omics don't make sense. So the ultimate goal is to improve outcome by personalizing therapies based on the unique characteristics that each patient's cancer presents. So if we look at the bedrock of all precision oncology, it's drugs, the drugs that are available to us. And the drugs that are available to us currently are a lot of targeted therapies. So this is showing, this graph is showing all the drugs that have been available to us since the 2000 or have been approved by the Food and Drug Administration in the US. And in Canada, we generally follow the FDA in the US. And then Health Canada would approve the drug based on recommendations from FDA and our own publicly funded healthcare system. Nuances here. So as you can see from this graph, this plot, is that almost 50% of the drugs that have been approved since 2000, the year 2000, are targeted therapies. So here you can see in yellow, there are all the targeted therapies. There are different kinds of targeted therapies we will get into in the next slide. But cytotoxic drugs, which are chemotherapies, form only around 7% or 8% of all drugs that have been approved since the year 2000. The rest are targeted biologics. Again, these are antibodies or humanized antibodies, like monoclonal antibodies against PD-1, PD-L1, which are called immunotherapies. So almost 90% of drugs have a biomarker associated with it, or they are targeted therapies. So these are, again, the targeted therapies that have been approved based on class. So this is so kinase inhibitors, or Dr. Nohr talked about this a little bit. These are mainly inhibitors of enzymes that are kinases or phosphorylated enzymes. These form the most common drugs in cancer, because a lot of signaling cascades are made up of kinase or are orchestrated by kinases. Then there are immune checkpoint inhibitors. These are immunotherapies that are being used currently in the field and uses the patient's own immune system, which is generally blocked by the cancer, and if the cancer tries to evade the immune system, these immune checkpoint inhibitors actually are antibodies that bind to these blockers, and they open up the immune system in order for them to, now the immune cells, to target the cancer and eradicate it. And then there are other agents here, which I'll not get into, but they are mainly agents like PARP inhibitors or anti-androgen therapy, but they all are against a particular signaling mechanism within cancer, or a particular target in cancer. All of these therapies are essentially approved. Most of them are approved for a certain cancer type. But gradually what we are now seeing is that there are more tumor agnostic or tissue agnostic therapies in cancer that are being approved. These are therapies that can actually act pan-cancer. What is important to understand here is that gene expression is mainly tissue specific. So usually a particular type of mutation, if you see in a cancer type, it will work differently in another cancer type because the expression of that gene in the cancer type might not be relevant to the development of that cancer. But there are certain genes that act universally, or mutations that act universally, like BRAF V600E. And these are mutations that are found in the BRAF protein. And this is a RAF protein, and essentially can act across different cancers like thyroid, lung, skin cancer. And there is this drug called vemurafenib, which can be used in different cancer types, including lung, thyroid, and skin, and has been approved by FDA as such for pan-cancer therapy in these cancers. And how are these drugs approved? These are drugs that are approved based on clinical trial data. So clinical trials are essentially experiments which are done on cancer patients, and for tumor agnostic trials, they are called basket trials, where all of these tumors like tumor A, tumor B, and tumor C, they're put in the same basket based on the mutation profile of these tumors. So if the patient has, or if patient A with tumor type A has a BRAF mutation, and patient B and C also have the same mutation, but in a different tumor type, like lung or thyroid, then they would be put in the same basket and be treated with the same drug. And that's what a basket trial is, because their patients are been into baskets based on their companion diagnostic or the targeted agent that the patient will be given. Again, these are some of the tumor agnostic FDA approvals that have happened since the year 2017 almost. So pembrolizumab, which is an ICI or immune checkpoint blockade, is now approved for microsatellite instability high tumors, as well as mismatch repair deficient tumors. Then there is larotrectinib, which is an NTRK inhibitor, which NTRK1, NTRK2, and NTRK3 form fusion proteins within tumors. These are found across tumors, and they actually were approved by the FDA for pan-tumor application in November 2018. Then also entrectinib, again, another NTRK fusion drug. This is entrectinib is sold by Bayer. Larotrectinib, I think, is from Roche. There are two different drugs from two different companies, but targeting the same population of patients with NTRK fusion tumors. Driven tumors. Then there is pembrolizumab, again, was approved in June 2020 for tumor mutation burden high cancers. So tumor mutation burden is a biomarker for immunotherapy response, because if you have high tumor burden, tumor mutation burden, then there would be new antigens that would be presented by MHC or class 1 and class 2 on the surface of your tumors. And that would lead to more recognition by immune cells, and immune checkpoint inhibitors would act better in that background. Same thing with dostarlimab, which is another immune checkpoint inhibitor, and acts the same way in DNA damage mismatch repair deficient patients, as well as microsatellite high patients. And we also talked about dabrafenib, where for BRAF V600E mutant tumors across cancers, and another RET fusion positive cancers would be treated by selpercatinib, which is another drug which is against this RET fusion. Again, this is a kinase, so this is a kinase inhibitor, but with pan-cancer efficacy. So how did we reach this stage? Like, how did we reach a stage where we had these particular biomarkers based on which we are now able to treat patients? We reached here because there have been several large-scale genomic projects that have generated a wealth of knowledge for genomes across cancer types, for transcriptomes, for epigenomes, and then proteomes, all of this data has been generated in order for us to understand essentially what kind of targets exist within cancers, what cancers are driven by what proteins or genes, and then are we, if you manipulate those genes, are we able to target them? So some of these large-scale genomic projects, we talk about this in more detail in our course, the Precision Oncology course. Both Dr. Nohr and I do that. But here I'm giving you a bird's eye view of what those projects are, like the Cancer Genome Atlas, the CPTAC or Cancer Proteomics Tumor Analysis Consortium. GTEx is unique in the sense that they have sequenced normal tissue for transcriptomics from around 1,000 individuals, normal organs across the body. And this sort of acts as a comparison of what has changed in cancer. So we always use this data as sort of a baseline based on which we ask the question that based on normal tissue expression, how does cancer tissue expression looks like, and GTEx is really valuable to do that. Then there is ARGO, or the International Cancer Genome Consortium, ICGC, which is again, like TCGA, has sequenced a variety of different tumors. There's PCOG, which is the pan-cancer analysis of whole genomes. This is again whole genome sequencing of a variety of different cancers across different tissues. Then there is the two cell line databases, Cancer Cell Line Encyclopedia and Genomics of Drug Sensitivity in Cancer. One is based out of Harvard and MIT, the Broad Institute of Harvard and MIT, the Cancer Cell Line Encyclopedia is, and the Genomics of Drug Sensitivity in Cancer is based out of the Sanger Center in the UK. And these are databases of cell lines that have been treated with a variety of different drugs. And before that, they have been sequenced, so you can then associate drug sensitivity to gene expression in cancer and tissue, and also mutations in cancer. So these are mainly pharmacogenomic analysis of cancer cell lines. One of the large-scale genomic projects that is from Canada is a very pioneering project that was started in the BC Cancer Agency Genome Sciences Center, Canada's Michael Smith Genome Sciences Center, where I actually trained. This is called the POG program, or the Personalized Oncogenomics program. I was an analyst on this program. This is a program where you take recurrent metastatic cancer patients and genome sequence them and transcriptome sequence them, also, as well as we do proteomics and now methylomics, and ask the question that what is altered in these patients' tumors so that they can be treated with a particular drug based on their genomics. This is a very unique platform where a lot of novel drugs are used, drugs that are otherwise would not be available for the patient, but based on their genomics, there might be. So one of the famous stories from this project was that there was this patient with, I think, colorectal cancer who had FOSS-Jun mutation, and Jun is a protein that is also altered in heart disease. And there is no Jun inhibitor that is available in cancer, but there was this drug that is being used for heart disease that was also targets Jun. And then there was this tumor that upregulated Jun, was treated with this Jun inhibitor from cardiac sciences, and it actually, the patient went into remission. This was a big story. And now more and more patients are being treated by these novel drugs in this particular program. So again, what are the different omics that are used in cancer? So we use transcriptomics, genomics. Dr. Nohr also talked about this, so I'll not go into more detail. So genomics is basically the sequencing the DNA. Proteomics is the protein. Transcriptomics, again, is the RNA. Epigenomics, again, is basically promotes a methylation and then other acetylation and stuff that is used for mainly understanding what kind of epigenomic alteration. These are alterations that are heritable but not directly DNA changes. These are mostly we look at methylation of the promoter region of certain genes. Then also there is metabolomics, which is looking at the metabolites and looking at what metabolites are secreted by the tumor. So we have already gone through a large-scale genomic projects, but then all of that information has now been distilled into what are driver genes in cancer. So based on that, we have separated out these genes and put them on panels. So we now sequence only certain targeted panels or targeted genes, which are drivers in a variety of different cancers. So we now use comprehensive genomic profiling, which is CGP, which is basically around 400 to 500 genes that we sequence, not the whole genome, because we already know that what are the genes that might be relevant to a particular person's cancer. So targeted sequencing focuses again on specific genes. It detects mutations. It can detect RNA changes or gene expression changes. It also classifies cancers into subtypes and can predict prognosis or therapeutic efficacy of drugs. The advantage is, rather than sequencing the whole genome, you are sequencing a small number of genes, so it is more cost effective. You can also go to a higher depth of coverage. That means when we do whole genome sequencing or any kind of sequencing, we sequence the same region of the genome iteratively many times. Usually whole genome sequencing is done at 100x. But here, when you're doing only a small number of genes, we can go up to 1000x or more, 2000x. In the case of liquid biopsies, which we will talk about, we go to 10,000x. And this has increased sensitivity than a whole exome sequencing. So essentially, why do we do this? And when we see a variant, is there a meaning or a method to this madness? So what can we see? We can see oncogenes. We can see oncogenes being upregulated. We can see tumor suppressors being downregulated by loss of function mutations. We see activating mutations in oncogenes. These mutations can be either pathogenic, or they can be variants of uncertain significance. They can be benign. They can be, they can be prognostic. They can be therapeutic. All of these variants, they have to be classified. And then there are professional guidelines that actually classify them. So there are specific variant classification guidelines that are available. These are, we follow the Association for Molecular Pathology, ASCO, and CAP guidelines. And these are classifications on the extent of clinical actionability, whether they are therapeutic, diagnostic, or prognostic. They classify the somatic variants by tiers. We will talk about those tiers a little bit. And these are based primarily on the variants based on published evidence from different studies that have been published already. These are population-based studies. And then there are guidelines that are associated with these studies. So what are these guidelines? So and then how do they classify the variants? So these variants are classified into tier 1, tier 2, tier 3, and tier 4. So tier 1 are variants of strong clinical significance, and they are therapeutic, prognostic, or diagnostic. These are level A evidence. So either there is level A or level B evidence. So either there is an FDA-approved drug, or there are well-powered studies with consensus from experts that would say that, okay, if this is present, this variant is present, there is a drug associated with it, or you can treat with a certain drug or a certain way. And then there is variants of tier 2 significance or tier 2 variants, and these are also therapeutic, prognostic, and diagnostic, but these are level C variants. So FDA-approved therapies are present for other tumor types, but not published for this particular tumor type, but there are multiple small studies that have been published. So then there is level C evidence. If there are preclinical trials that are available, like in mouse or others, that would be level D evidence. So a lot of reports would say that. And then there are tier 3, which are, again, not observed at a significant allelic frequency in the general subpopulations databases, or pan-cancer and pan-tumor, and no published data is available. So these are variants of unknown clinical significance, and we don't really know what to do with them. But then again, we might be able to, based on consensus from experts, if there is no other option for the patient, sometimes there is a Hail Mary, and people try treating the patients with drugs which are targeting variants of unknown significance. Then finally, we also have tier 4 variants, which are basically benign variants. These are not linked to cancer really, or there are observed significant allelic frequencies in subpopulations. These are in normal databases like the 1000 Genome Project and others. These variants have been found. So they are really benign variants, but they are not associated with cancer. And we would classify them as such. These are some of the knowledge bases that once we generate our whole genome sequencing or even targeted panel data, we would go and query these databases like Civic, OncoKB, and others in order for us to understand what are the variants of significance in this cancer patient, and how do you stratify them. Quickly, I'll get into, we got into targeted panels. So we also want to talk about a little bit about a newer technique, which is called circulating tumor DNA profiling. This is, again, targeted profiling. It can also be done across the whole genome at low pass, which is whole genome ctDNA. But usually in the context of therapy and patient management, currently what is available is targeted ctDNA profiling. So this is circulating tumor DNA. This consists of small fragments of DNA that are released by tumor cells when they apoptose into the bloodstream, and they are small fragments representing the tumor, so mutations that may be found in the tumor. And these are essentially minimally invasive. So you draw blood from a patient, separate out the plasma, and that's how you sequence the circulating tumor DNA in the patient. It is the same as finding mutation in tissue. This is just sequenced at a much higher depth because the number of normal molecules, DNA molecules, swarms the number of tumor molecules that will be found in blood. So you have to sequence them at a much higher depth. So what are the applications of ctDNA? Again, early detection or screening, because if you can't see the tumor, you cannot really sequence the actual tissue. You can sequence the ctDNA, and that can lead to early detection of the tumor. Then there is treatment monitoring or minimal residual disease monitoring. This is done real time by serial blood collections and doing ctDNA analysis on these to see when a patient's cancer might come back after therapy. Then there is also predicting relapse, so early identification of molecular relapse before actually clinical relapse happens. That is done usually by imaging or CT. Here you would serially or longitudinally get blood samples from the patient, and when circulating tumor DNA appears, which might be much earlier than radiographic relapse, that would basically be used for treatment decision making at that time, or further patient management at that time. I would like to end by essentially what the future looks like. And everywhere, as you can see nowadays, artificial intelligence has taken over our lives, and we are essentially using artificial intelligence in our day-to-day lives. ChatGPT, people use ChatGPT all the time. Those are generative AI models, and the precision oncology is no stranger to the use of AI. And this is mainly relevant because we have had an explosion of data in cancer, based on the genomic data that we have generated, all the clinical trial data, imaging data. So we have got CT scans, we have got slides, which are H&E stain slides. These are routine slides that are used to diagnose patients with cancer. Then there is also the complexity of the disease itself. So if you can use AI in order for us to analyze multiple data types that are being generated using all of these complex techniques, it would really help. And it is really beyond human capabilities to do that in a very effective manner. So AI is being summoned to do analysis for this kind of complex analysis. Then also AI accelerates the process of finding and identifying different biomarkers. Not only is AI being now used to identify biomarkers, but also AI is being used to develop new therapies for patients. So, and also for early diagnosis. So these are the key applications for AI currently in precision oncology. Genomic data analysis. This is, again, very intuitive because we are generating large amount of data. To integrate all of the data and find a method in this madness, we need AI. A lot of medical imaging data has been generated. There is deep learning is being used in many labs, including mine, where we are trying to ask the question that can we find features in these images that can be used for diagnosis as well as prognostication. Then there is also the AI in drug discovery and development, where we are using AI for drug screening. So recently, we were able to screen 3 million molecules for binding to a particular protein that we identified to be an oncogene in a particular cancer type. So you need huge computational resources for that. But we are now able to do all of that. Before, we would basically look for physical binding using wet bench experiments that would take decades. Now we can screen millions of compounds within days against certain targets in tumors. Also, we have predictive modeling of outcomes. So AI can be used to predict recurrences, survivals, as well as treatment outcomes in patients. So these are some of the AI-powered tools that we use. So IBM Watson is currently being used in oncology. So this is IBM Watson for oncology. Then there are big companies in the US like Tempus, who are using AI to guide personalized therapeutic decision-making in cancer. Then there is Path AI, which is another platform where you can take slide images and query them for, again, prognostication and other biomarker identification in cancer. We already talked about OncoKB and CBio portal. This provides oncologists with AI-assisted insights into cancer mutations and clinical trial data. So all of this is now being used to generate better outcomes for our patients. And moving on, essentially, this would be sort of the future of precision oncology. And as we get into more and more complex data types, the idea would be to simplify our approach of distilling all of that data into bite-sized nuggets that can be used by clinicians in order to treat patients. So this is, again, this is the future of AI in precision oncology. So again, integration of multi-omic data, real-time monitoring and predictive analytics, personalized AI, where we will develop models based on, these are called living models, where we basically can develop models based and then change them as we generate more data on the patient. And then again, there are low resource countries where AI might be a very good fit in expanding the reach for precision oncology in these countries. So with that, I'd like to thank all of you We have recently moved into both Dr. Nohr and I have moved into the new Arthur Child Cancer Center. So this is sort of a rendering of that. We are on the 8th floor of this building. And here we are advancing precision oncology with being able to do research as well as patient care under one roof for the first time in Calgary. So with that, I would finish here and happy to answer any questions you might have.
So thank you so much, Pinaki. So we managed to gather a couple of questions in the chat. So maybe to start, we don't have much time for questions. So first question is for Dr. Nohr. The question is regarding to essentially access to resources and treatments in rural areas.
Sure. So just to be clear, I don't treat patients. I'm a molecular pathologist, so I work in the lab looking at cancer tissue samples under the microscope and overseeing testing. So I can't really talk directly about access to treatment. But in terms of access to lab testing, we are fortunate in Alberta to have a provincial molecular pathology program that administers all the publicly funded molecular testing. And so whenever we develop a new test, we develop it for all Alberta cancer patients. Now, it is centralized at our two major centers in Calgary and Edmonton. So patients in rural communities, what would happen is they'd have samples collected maybe over there and looked at by a pathologist in that setting. And then the tissue sample would be sent to one of our centralized labs for clinical testing. So there should be access to everyone. Of course, there's always hurdles sending samples around, but it is available.
Great. And Pinaki, there was another question in terms of the selpercatinib and whether it's used as a first-line treatment or a second-line treatment.
Second line, right now it is used as second-line treatment, I think. So it's not first line yet. Some of the like NTRK fusion based tumors, those are in the first line. But selpercatinib is not actually first line yet.
All right. There are a couple more questions in the chat, but maybe I take a question from Malcolm.
This is maybe a question to start with Pinaki. You mentioned you were using some algorithms to screen targets, presumably to find binding molecules to a kind of target sequence or to a specific protein. And this is kind of a tech transfer office focused question, but what is the plan for that data, or what is kind of the results in your world if you are to get a good hit on your current screens? Where do you go with that data, and what's the plan?
Oh, so you're talking about research, not actually patient treatment then?
Yes.
So within our lab, we do a lot of analysis. My lab is half genomics and bioinformatics and half wet bench. So essentially what we try to do is we try to find associations in the genomic data and then try to ask functional questions around that. So based on that, we were able to discover from large-scale genomic analysis of amplifications in our genome, in cancer genomes, especially squamous cell carcinomas. That's what we looked at. We found a particular particular protein that when upregulated acts as an oncogene. And that we were able to then screen for different drugs. And we have gone through that path of drug development, which was very, very, and I am not a chemist, so we are getting other people to help there, and we are building a team. So essentially, the idea would be to patent something. You cannot patent a gene, a target. You can patent a drug against that target. So the idea would be when we have enough data that a particular molecule or small molecule inhibitor of this drug or of this protein actually works, we will then try to modify it in order for, you cannot just patent something that is already available. So you have to modify it, make it more effective, hopefully, and then go on to patent it And it's a long road after that.
Absolutely.
Clinical trials and then,
Yeah.
all of that. So we are at a very early stage. So there's not yet anywhere close to doing anything with patenting yet.
Okay. Well, that's good to hear. We'll have to chat soon then.
Absolutely.
Thank you.
All right. There's a few more questions, but I'm really not sure if we have enough time to address any of these. Maybe try one more by Lisa. What are some ways that you think providers and our patients could stay informed about the latest options for personalized treatments?
Yeah. So in terms of providers, like for medical oncologists, for example, to be aware of the latest available options, they have their professional meetings, they have contacts with pharmacy to learn about things coming down the pipeline. They have discussions among themselves as colleagues. So they're continually, keeping an ear to the ground and keeping abreast of the latest options. And then I think really for patients that want to stay informed, the best thing really is to talk to your oncologist because they should be keeping up to date with that. And there's a lot of nuances to like what treatments are available that are publicly funded or maybe available through patient pay, and what can be done that could be done on like a compassionate access for things that are off-label or through like specific approval processes. So there's a lot of nuance and complexity there that the oncologist should be involved in helping to navigate to determine what is the best treatment plan. Ultimately, the treatment plan as well depends on a synthesis of the patient's whole presentation and disease status and their other comorbidities. So that really requires that medical judgment and that conversation.
All right. Thank you so much to both of you and to questions, and apologies that we didn't get to all the questions. But I think if there are any burning questions, you're welcome to also email Dr. Bosse and Dr. Nohr again further insight. All right. Ivy, any last comments?
No, just thank you so much. Thank you for presenting, Erik, Pinaki, Maya. We really appreciate your time and expertise. And thank you everybody for joining. Please feel free to check out the other events this week and feel free to apply for any of the program options or courses that we have available. So have a great rest of your Monday. Stay warm out there and we hope to see you soon. Take care. Bye-bye.
See you then.
Bye, everyone.
Bye. Thank you.